This is episode 295 of the Stem Cell Podcast, Roadmap for Cell Therapy Development with Doctors Jacqueline Barry, Kapil Bharti, and Jack Mosher. Hello, everybody. This is Dr. Daylon James with Dr. Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you're enjoying the listen, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have a triple threat. Doctors Jacqueline Barry, Kapil Bharti, and Jack Mosher. The three of them are on the podcast to talk about ISSCR's best practices for the development of pluripotent stem cell-derived cellular therapies. This is kind of the phase two of that phase one standards or best practices on the research, although this has much more translational bent, which is appropriate given the exciting times we're in. We've also got our usual roundup of recent highlights and stem cell news that's coming right up.
But first, ISSCR 2025 is almost here. Dr. Barry will be chairing a session on the best practices for the translation of pluripotent stem cell-derived therapies on Thursday, June 12th from 8:30 to 10 o'clock a.m. Hong Kong time. So if you're attending ISSCR 2025, be sure to check it out. And you can find the full program at isscr2025.org. We're going to start things off with not a pure stem cell story. It's utilizing tools that, of course, we all use in stem cell biology, including CRISPR. This is actually you probably have heard about this. This is a New England Journal of Medicine article that was titled Personalized Gene Editing to Treat an Inborn Error of Metabolism. And we wanted to highlight this just because this is really an incredible milestone in the genome editing field, and we do cover genome editing papers every once in a while on the show, on the translational side of things, but also on the tech development side of things. This is work that's led by Kiran Musunuru, who is an amazing cardiovascular biologist. I've got the opportunity to know him over the years. He's at UPenn, previously was at Verve Therapeutics in Boston, and is one of the world leaders when it comes to utilizing CRISPR for in vivo gene editing. And this was the first example of a patient-specific in vivo gene editing approach to treat a rare genetic disease. And just the story behind this is so incredible. There's a kid whose name was KJ. He had this really debilitating metabolic disease that's called severe carbamoyl phosphate synthase 1 deficiency. Very specific disease, rare disease. Importantly, it has an estimated 50% mortality in early infancy. So after KJ was born, the docs noticed this problem, in terms of his metabolism, and, he was having some really severe symptoms. So they needed to act very quickly to develop something that may be able to make his life better or potentially even save his life. So what Musunuru and his collaborators here, and of course, this is a very collaborative story of more than 40 authors here. And last author is Rebecca Ahrens-Nicholas, also at UPenn, I believe. What they did was they started collaborating with a bunch of folks to develop a customized lipid nanoparticle-based delivered base editing therapy. So base editing, again, a specialized version of CRISPR that was, developed in part by folks at the Broad Institute. A more targeted approach for genome editing, right? So they developed that in collaboration with the industry colleagues, clinical colleagues, and then also obtained a rapid regulatory approval for the therapy. Skipping obviously a few months here, but the patient, this kid named KJ, he received 2 infusions of this gene base editing approach to correct this mutation around 7 and 8 months of age. And in the 7 weeks after the first infusion of this base editor, He was actually able to have an increased amount of dietary protein, a reduced dose of a nitrogen scavenger medication. So his medications went down to half the starting dose. Importantly, reduction in adverse events, a few viral illnesses. They mentioned no severe adverse events, but certainly need a much longer follow-up here to actually warrant the safety and efficacy of this approach. But again, an incredible milestone for this CPS1 deficiency, which is, again, an ultra rare inborn error of metabolism affecting specifically the urea cycle. In terms of the numbers, it affects 1 in 1.3 million people, 50% mortality. Liver transplantation is the alternative usually, and this is something perhaps a bit more palatable when it comes to this in vivo editing approach. So, yeah, I mean, I don't want to go super deep into it. It's not truly a stem cell story, but still a significant milestone. We're talking about using genome editing in vivo to fix a very specific mutation and potentially save a kid's life within the first year of life. And you can imagine how this is an approach that's going to be expanded across the board to other rare diseases. And it's something that Dr. Masnuru is really interested in working on. He's one of the folks who actually initially worked on some of the PCSK9 cholesterol regulating approaches as well when it comes to genome editing. And I think Verve Therapeutics is still really actively involved with that. But really incredible here. I think a lot of this was done free of charge because this was such a cutting edge treatment and was such a critical life-saving approach that they had to do for this kid. So, down the road, you got to think about the economics of developing an approach like this because we talk about this on the IPS side of things, right? I mean, yeah, the dream is you have personalized medicine for everybody, and that's what this was. It was a personalized genome editing for this kid. But that's expensive, and economies of scale and all that. We got to think about that down the road, and that's part of the reason why people in the stem cell field are focusing more on autologous in general. Part of the reason, but regardless, just a really great milestone here, helping to potentially save the life of this kid with CPS1 deficiency. And I'm excited to see this work being extended to other rare metabolic diseases and rare genetic diseases as well.
Yeah, that last point there. I mean, you started by talking about this is not exactly a stem cell story, and it's not exactly a stem cell story. You can make the argument anytime you're targeting an organ with regenerative potential to do a genetic fix, it's going to take some stem progenitors to amplify and like lead to a cure, which is what this hopefully is. But it's more than a stem cell story, and it is, in fact, a stem cell story in the abstract. And bear with me here because for me, the real takeaway, the science is amazing, but I think we all collectively, the scientists as well as I think the lay public, has a deeper understanding of, the tech here, how it worked. We've talked about it a lot, as you said. But for me, this is a sea change and a watershed moment in terms of the deployment of this tech, right? Because this is a proof of principle, not of the tech itself, although it's that as well. And again, fingers crossed that this is a lasting cure for this kid. Um, but it's like a proof of principle of the apparatus that it takes in order to deploy something here on an individualized level. And sure, like this was very expensive, of course. And of course, it was free, that it was very expensive, both in terms of, actual resources as well as effort, human resources and anguish on the part of many of the players, because this was really first, not first necessarily, but among the first of its kind. But they did it, right? They did it. The regulatory apparatus, I mean, compassionate use, sure, but unlike all the other compassionate use that we talk about, typically this is like, you're expecting the patient's demise. This is a potential cure, right? So the proof of principle that you can do it, economy aside, but you can mobilize in the time it takes. And notably, I think this is interesting to me, fascinating. Of 45 authors, 12 of the authors on this paper were from IDT, from Integrated DNA Technologies. And that just shows you there was a bunch of scrambling here just making oligos. So, and massive effort towards that. I'm glad they got the credit because this was such a heavy lift, but it was so worth it. One life is worth infinity. But eh, I think this is it. This is a proof of principle that this is one case, but there's a lot of unique diseases that could be treated in this same way. And God forbid if this had been a fail, or if let's hope it doesn't turn out to be diminished returns. But the fact that it's a success, I think, brings a lot of attention. Poor Kieran is going to be his going to be lighting up with every parent of a sick kid, monogenic diseases at least, looking for a cure. But that's, I think, a good thing is that there's an option, there's hope, but also there's a reality of seeing how widely this can be deployed. You can't necessarily scale this thing, but you can scale the approach to bring individualized cures down to a reasonable price. And I think that's the road that we're set on now, and it's so exciting.
Yeah, incredibly exciting, and I'm glad you mentioned IDT. Yeah, because I was reading some of the press articles on this particular, amazing approach, and they were saying that yes, there was free labor, basically donated labor from multiple industry partners, including Integrated DNA Technologies, IDT, Aldebaran, Aquitas Therapeutics. These folks were not in this study to make the money, right? They were doing this for the kid to really uh, uh, make sure the kid was okay. And that's an incredible thing to see in this age of course, $1 million treatments and all this. And of course, that's down the road. The company's got to make money off the therapies. But for right now, what's most important is this kid KJ, that he's getting better. Hopefully, that he had a cure that's long lasting. And I'm just curious about the follow-up. I hope this kid stays really well. Obviously, as a parent of two young boys, this hits really close to home for me. So, great job to everybody involved with this amazing study.
Yeah, guys, read the piece in the Times if you haven't already, which I'm sure most of you have, because it really does give you hope. Even as someone who's aware of the possibility, this really was awesome to read. And from a like a practical standpoint, I hate to bring it back to the science, but this is unlike the unregulated stuff that has been done in the past with babies, and you all know what I'm talking about. This is a patient that we can follow up, as you just said. Are you waiting to see the follow up? Well, this is a living experiment, one among many future living, I don't want to call it experiment. This is a life saved, but the value to science and the multiplicity of lives saved using this and follow-up results from this patient is going to be truly immeasurable value. So can't really talk about this one enough. And here's my segue. This is a effort that was really mobilized very much in terms of in silico. We live in this age of in silico medicine where the formulations and the therapies are developed all in this nether space. And I got a story here from Nature Methods that's kind of giving a similar kind of matrix overlay to our physical world. This in this case, focusing on organoids, right? And we know the story with organoids, we've been talking about them for a decade now. And the big innovation there was the 3D, right? In order to really understand biology of cells and tissues, you got to understand 3D, right? Not just biology, but pathology, of course. And organoids and, of course, the organ on chip systems have emerged as these really critical tools because they're a 3D assembly, right? That more closely emulates the both the architecture as well as the functional capacity of these basic, and I want to emphasize that basic organ units. We're not yet there where we're getting to the complex architecture of organs themselves, but really kind of the primordia and the elemental functional elements of those organs. Um, and of course, they're the physiological relevance of these organoids is greater, but there's more to be done. And a lot of the efforts nowadays are using these bioengineered matrices and micro niches with, orthogonal, morphogens, all that we talked about just last episode to enable the precise control, not just of the differentiation of these cells, but the geometry and the architecture as well and the patterning, right? We just talk about Lutoff a lot because he's blowing up, but those crypts, right? You need to kind of enforce some kind of architecture and let it be grown into in that case. Um, but the studies to date have focused more on a like gross level or overall organoid morphology as a measure. Also, gene expression, ton of single cell to validate and immunostaining, all these kind of static definitions that qualify these things as physiologically relevant. Uh, but what we want to move towards, and this is the story I'm talking about, is a more complex morphological characterization. So you can really understand at the cell scale what's going on, see how reproducible these organoids are, and also deploy it in these kind of high content screens, right? Combining the 3D component with the content screening component, which remains a challenge today. It's much easier in monolayer culture for obvious reasons. Um, but there have been some approaches and some software developments for these deep learning based solutions to meet the demand for this 3D cell segmentation approach. For example, there's this organoid tracker, but that's just tracking nuclei alone. And a lot of the techs, there's a lot. I'm not going to name them all, but many solutions have been offered up. But a lot of them require really high quality 3D image resolution that isn't really practical in real world conditions. When you look at like real world in the lab or clinically, you have lower resolution or poor signal to noise ratio. Um, and also, a lot of these methods, while they can be practical and give beautiful results, there's not real clarity on the computational time and or demand, right? Do you need a supercomputer and does it take weeks? Like the practicality of these things for high throughput applications, like the screening, the high content screening that I talked about, remains to be seen. So in order to address these limitations, a group comprised of many investigators that are known as the OrgaRes Consortium, which was led by Victor Racine at Quanticell in Montpellier, France, and Anne Begging, who is at the National University of Singapore. They introduced this new methodology for assessing the 3D morphology and topology within organoids by generating these tailored algorithm. This is super tech, so I know I may be losing you, but the application is it. They got these tailored ag algorithms for multi-scale. I didn't know what this meant either. Multi-scale segmentation and quantification, and this is a key of the nuclei as well as cytoplasm. And then whole organoid level. So you're going down to the nuclei, also clusters of cells, also the whole organoid. So this is kind of multi-scale. And all you need is markers of nuclei and plasma membrane, super basic. So you don't need, complex immunostaining or anything. And then they use these proof of concept case use experiments with either well characterized condition. Here, talking about osmotic stress in, I think, hypotonic, so some tonic solution. And also to deploy, they got results with consistent with what has been described. And also they deployed it in these kind of unknown use cases to try and illustrate the capacity for discovery here. And Arun, you'll appreciate this. They looked at mechanical stressors like microgravity to see how that this multi-scale 3D cell segmentation analysis work in that context. I got very interesting results that I didn't really understand. Arun, you should have a look. Um, but the key here is that you don't need the labor-intensive immunostaining. You don't need some advanced computing. You don't need to be a programmer. So this is accessible to standard, quote, standard laboratory setups, although I will, point towards the fact that you need some pretty high resolution, Z-stack type imaging here. We're talking about like light sheet. You can't just get on your epifluorescent microscope and plug it in to their program here. But that's to that note, they have this really user-friendly software interface that they developed as well called 3D Cell Scope, which allows any researcher. So you take your legacy images, plug them in here, and get some new insights, right? So I like any kind of tech tool. Nature Methods story that has a relevance to organoids. For me, it's like guaranteed that within 10 years you're going to have 1,000 citations. So I want to highlight it, Arun, and I got the web address here, and you can see it posted on the Stem Cell Podcast website because I can't wait to get in there with some of my old school Z stacks and just get a new view.
Cool tool. Absolutely. Always a fan of folks who make these really nice repositories and online websites and tools for all of us to play around with. I didn't realize this was a microgravity story, so you caught my attention now, Dale. And hey, thanks for that.
You got it.
Hey, I appreciate that. I actually had to dive into that a little bit more and see what sort of experiments they're actually doing in microgravity to validate some of this work because as somebody who does stuff in space, there's all different kinds of microgravity that you can subject cell cultures to. There's simulated microgravity here on Earth, where you can just put these cells and spheroids and random positioning machines, and they kind of generate random force vectors and sort of simulates microgravity in that way. You've got magnetic levitation approaches, random suspension bioreactors, which is not perfect. I mean, I'm biased, but I think the only true microgravity out there is in space, where you don't really have force vectors going all over the place, right? Here, they actually use a form of simulated microgravity called parabolic flight. So you might be familiar with this,
The
the
Vomit
the
Comet.
Vomit Comet.
Yeah.
Exactly. So they went on to the Swiss parabolic flight program, went on a Cessna, one of the vomit comets over there and somewhere above the beautiful Alps, and then subjected these spheroids to microgravity, free fall basically for about 15 seconds on that parabolic trajectory. So it's it's not the best model of microgravity, but it sure is fun. I would love to go on I would love to go on the Vomit Comet sometime. But hey, it's a really cool data set. And I'm excited to mess around with it as well.
That is so dope. Is that in like the materials and methods?
Yep.
That
Yep.
is awesome.
It's very cool.
I wonder how they slipped that into like the funding proposition there, but I justified. You're better there than going up into space. And you would know, Arun, and I'm very pleased and proud to share that by the time this episode airs, you will have been announced as the director of the new Center for Space Medicine Research at Cedars, where you guys are using, low Earth orbit to explore the impact of spaceflight on the human body and biomanufacturing applications. So, very proud of you, my man. You nailed it. You're going to be the first scientist that I know who's going to space. So, take a picture for me.
Will do. Hey, maybe you can come up there with me. I don't know.
Oh, I
I'm
don't
sure
think
it'd
I'm
be
prepared for that. I'm
no.
too lanky.
I don't think anybody's prepared for it, but actually, I appreciate that. I really appreciate that. It's been in the works for a little while, so it's fun to highlight another microgravity study here. And actually, the keynote speaker, the first speaker for our new Center for Space Medicine Research here at Cedars is going to be Kate Rubins, who is a former guest of the podcast, friend of the show. So, yeah, a lot of exciting things happening on and off Earth here in LA. 250 miles up. That's how far up the space station is, just FYI. So, moving on to stuff that's more relevant to those of us here on Earth. And, we're talking about organoids again. It's kind of another organoid story here, but this is a more developmentally oriented organoid story. It's a really exciting cell paper. Coming from the lab of Sylvia Santos at the Crick, Francis Crick Institute in the UK. Quantitative stem cell biology. Title of this paper is Post-Gastrulation Amnioids as a Stem Cell Model of Human Extraembryonic Development. Just another amazing oid, developmental organoid here. unraveling that black box of human development. And we've been talking about these so often here on the show, these various models that are emerging from leaders in the field. Like, we've had Jacob Hanna, we've discussed Magdalena Zernica-Goetz's work. I'll talked to Alfonso Martinez Arias, various leaders in the field working in this area, and I'm sure this is going to be a hot topic at ISSCR as well, these early models of human development. But here, it's a new model system looking at the development of certain extraembryonic tissue, and in particular, the post-gastrulation amnion, right? So the amnion, as you might know from developmental biology, is this extra embryonic tissue in mammalian embryos, and it's believed to provide this really critical signaling, structural, nutritional, important right there, nutritional support during pregnancy. Um, critically important, of course, but there aren't great models, as you might expect, to study human amnion formation. And the in vitro models are lacking, and certainly there are differences with mouse development and human development. So that's where we are right now. So they developed a embryonic stem cell derived 3D model of the post-gastrulation amnion. And the really cool part of it, it actually has like amniotic fluid that is developed as part of this organoid. I don't know if you remember a few years ago, we covered that story about like the organoids that develop the cerebral spinal fluid, where
Yes.
they just have the liquid in there. It's the same kind of thing. It's really wild when you can modulate development in vitro to get that same kind of fluid that you would get in vivo. It's really crazy. Um, so they dub these things post-gastrulation amnioids or PGAs that, in their words, faithfully recapitulate extraembryonic development up to 4 weeks post-fertilization, mimicking the functional traits of the human amniotic sac. These PGAs can self-organize, form the amnion and the yolk sac, again, extraembryonic tissue, and are surrounded by the extraembryonic mesoderm as kind of a part of the development here. They did some further understanding of the mechanism of how these things develop, and they showed that GATA3, a really critical transcription factor, is required and sufficient for amniogenesis, and that there's an autoregulatory feedback loop that actually governs the formation of the amnion, where the extraembryonic signals promote the specification of the amnion and the formation of that fluid as well. It's scalable, it's reproducible, which is why I love it. You can integrate it into your drug discovery and screening workflows and your high throughput workflows. It's got, yeah, like I said, reproducible. It's got this precise cellular, structural, functional integrity, and opens new doors for figuring out how the amnion and the embryo might be able to interact with each other during human development, beyond gastrulation. And really, I think pretty exciting stuff here for pharmacological studies down the road, and who knows, maybe even clinical applications as well. So really neat stuff here. Check out the pictures, check out the illustrations, just how that fluid is actually accumulating in those little sacs. It's really neat to see. Um, it's amazing what you can make in vitro these days, right, Dalen?
Amazing. I'm looking at it right now, and it's like the size of a grape. They got this video where they're shaking these little amnioids around in a flask, and they can only fit two of them in the flask. So, wow. And that, as you said in the jump, pretty much, is what's so neat about this, right? That functionality. And yeah, that was Lancaster when we were talking about the CSF, and that was such a home run, right? Because it's like it's so gratifying, right? When you can see a cell that's doing the thing and creating the substance that it does in the body, right? And here, because it's so near in early development, the amnion is formed so early, it really underscores the power of these cells to recapitulate like real biology and form real things. I know that seems like such a dumb thing to say because that's why we're all in it, right? But I think every now and then we need a reminder because we're all going after these tissues that are so complex and not to, least of which lack like the vascular system, not integrated. There's so many pieces missing that every now and then I really enjoy being reminded of how we're making cells that do things, and these cells, as you said, illustrated by the amniotic fluid they generated and the size, but also the molecular architecture and markers and the biology and mechanism that they elucidated there with the GATA3. This is all of it, and no coincidence. The big cell story. With any of these stories, I'm waiting to see how they integrate it. Like, is there going to be someone that does like trophectoderm, amnion, gastro, and tries to get all these things together into like a fetaloid? I hope not, quite honestly. But I always wonder what the end game is with the amnion, despite like the obvious mechanistic understanding and impact for early development, implantation, and etc. I wonder if there's a cell here on ex vivo or in vitro cell therapy derived from these amnions. We'll have to see.
We'll have to wait and see, but in the meantime, plenty to figure out mechanistically about development as well. Figuring out, like I said, the extra embryonic tissues, how they interact physically and mechanistically with the embryonic tissues. There are missing components, of course. There's like, the chorea and the embryo proper are not in this model, like you're alluding to. But yeah, I think the functionality is the fun part of a lot of these organoid models. We've talked about a number of these. Of course, you got this one with the amniotic fluid, you got the CSF model. I love the cardiac organoids because they beat just like the heart does in the body. But I don't know if you remember those ocular organoids, which apparently secrete
Mm-hmm.
tears, right? That's another example. And I was thinking about these while you're talking. Then there are also the hair organoids that actually have little hair follicles too. Um, and seeing is believing, but the functionality is kind of the fun part of it. It's almost like we're science fiction fans, and you're doing these like sci-fi like organoid preps in a dish that have real applications. Each of these things that I mentioned could have clinical translational applications, perhaps even for pharmacological screening too. So a lot of really exciting things that you can figure out just by making these little balls in a dish, right?
Big balls, Arun, not so little.
I wasn't gonna go there, but I guess I made you go there.
You just took it somewhere else. I was just adding a little scale there. All right, we're gonna finish up with a story that's a bit of a callback to a story we did a few episodes back, and I'll come back to that. But this is it's an Evo Devo thing, which I love ESLs for. Talk about Lancaster with the CSF. She also is a pioneer in using stem cells and organoids toward this end. It's a callback to that too. It's a callback. And of course, we're talking about human. And anytime you talk about anything, it's because we care about the humans, right? In this case, we're talking about our big old brains and our extraordinary cognitive capacities. Although I think the argument could be made that they're slipping amongst some of us, um, myself included. The whole big brain thing, right? And by virtue of the size, the enhanced cognitive capacity, it's about neural cell number, right? Also, though, the diversity and the circuitry. And the reason why we have those big brains that are highly connected with a lot of different types of cells is because we have this protracted period of corticogenesis. And that allows for increased neuron production, but also maturation. And this manifests with these abundance of upper layers, right? All that wrinkled brain. So that's where the magic happens. And during cortical neurogenesis, the primary progenitor, their neural progenitor cell that you have in the ventricular zone are these radial glial cells, the RGCs, which derive from neuroepithelial cells. And first, early stage in humans, they undergo self-renewing divisions to expand this kind of amplification pool, and then they shift to differentiation as along with dividing to produce neurons and basal progenitors. So in mice, uh, with their little brains versus humans and non-human primates, uh, it's not as robust, right? In humans, non-human primates, you have these RGCs and basal progenitors there are more of them, they're more diverse, and they grow more, right? So it's clear why, the correlative element there, but we don't really understand how uh, the these progenitors and their organization and division, et cetera, is controlled to generate these features that are unique to the primate and the human. What we do know, again, correlative studies with comparative genomics have identified a lot of human-specific loci, right? And not really specifically looking at non-human primates versus humans, there's not that many differences in like the actual coding sequences, right? We're very similar, 99 point whatever. But there's a lot of differences in the non-coding DNA. And notably, this is most notably illustrated by these human accelerated regions. And this is a callback in episode 289 with Robert Zweiger, which you should listen to independent of the Roundup story because that was just such a delightful interview. But we talked about these HARs a little bit. It was a story from Yin Shen's group at UCSF. And the idea there is that there's these human accelerated regions that, many of which are located near these neurodevelopmental genes that, in all likelihood, account for those differences in our big brains and our cognitive capacity. But putting aside exceptions like in the skin, in the developing limb, where these HARs are described and functionally described, the direct function of HARs in vivo is not well known, and the huge gap there is how literally a majority, I would argue, I don't know, we haven't found them all, but like thousands of these HARs contribute to the development of the human brain. But the understanding of that is pretty shallow. All right, enter Deborah Silver and her group at Duke University Medical Center. Go Duke!
Go Duke.
They previously discovered in the lab this HAR E5, right? Which is highly divergent between humans and chimps, and exhibits this species-specific enhancer activity in the mouse forebrain, right? And and many studies have shown that this HAR-E5 acts as an enhancer of frizzled 8, which is a Wnt receptor. So Wnt is clearly involved. Wnt is involved in cortical progenitor proliferation and differentiation and disease. So no coincidence that this HAR-E5 is involved in regulating, progenitor function in the brain. Now, here's the thing. If you have transgenic mice, which express the human HAR-E5, you get enlarged cortices, and you get mice with big brains. Get that. Crazy, huh? I don't know about the cognitive capacities. You should read the story. But again, the correlation is there, but how mechanistically that control is exerted and how it's specific to the human is unknown. So here we go. This is a massive deployment of all kinds of models across, they genomically edited mice. They also use human and chimpanzee cells to deconstruct the action of this HAR E5. And what they showed by overexpressing, so they used these mice that were genetically altered to overexpress this HAR E5, and they showed mechanistically, they went deep and showed that they have, these larger neocortices, and they have more excitatory neurons, right? And also, there's an increased functional independence, right? They do these in vivo functional dynamics, like looking at neural dynamics, and show that there is independence between these cortical regions. So they're kind of getting an idea of how the parallel processing that these larger brains are capable of. In addition to being larger, the circuitry is altered and expanded. Then they went deeper, showed that it specifically acts by modifying the RGC behavior by causing increased self-renewal in the early developmental stages. And then later, when there's differentiation, they have expanded neurogenic potential. So they grow more and then they differentiate more. So they're just more robust, juicier. And then going into the cell-based systems in the human and chimp, they made both neural progenitor cells and cortical organoids and showed that there's increased that activity is driving increased progenitor proliferation when they put it in the primate or human background. And here we go. mechanistically speaking, by amplifying canonical Wnt signaling. So as usual, everything comes down to Wnt. But what I love about this story is both, the parallel orthogonal approach using three models that are mammalian models that are highly relevant in the brain to kind of get that longitudinal developmental process, as well as breaking it down in the cells to manipulate systems and overlay them, and not just the human, but in the non-human primate here in the chimp. I thought was just such an elegant approach, and I think it's so sexy that this horror stuff, because finally the brain, which I've always thought was just so impenetrable, is being understood on a different level using this Evo Devo approach. So, one of the earliest and I would say most compelling applications of pluripotent stem cells is to understand our evolutionary origins, which is such a mind F, no pun intended.
I like what you did there. You know, this is awesome. I mean, really a amazing mechanistic deep dive here. Um, I didn't do a mechanistic deep dive into the story, Dalen. My apologies. But when you mentioned introducing this human element, this HARS element into mice and um into non-human primates, did they not do any behavioral studies? Or was this exclusively in vitro?
Like I said in this story, no, there was in vivo looking at the
Right,
mouse and more
right.
neurodevelopment, and I don't know, like behavioral, not so much behavioral, but more of the neuroimaging in order to look at activity. But I don't know if, well, in this story, I didn't see it, and I don't know if in the previous story, because this is built on a mouse where they overexpress, same thing, where they first observed the larger neocortices. And I would expect that that was the first question that reviewers asked there in terms of behavioral. So maybe have a look at that, but I do not have an answer for you. My apologies.
Yeah, no, I'm surprised because I was diving through the figures, and maybe it's in the supplement somewhere because that just from a big picture perspective, that's my biggest curiosity. And again, it's very hard to tease apart mouse behavior. I get it. As when you're modulating some of these, neurogenic factors, but I'm not gonna say what I'm thinking, but I think you can think what I'm thinking. Um, but I'm curious about the behavior of both of these animals after this modification. I'm not going to say Duke University is bringing you Planet of the Apes or Planet of the Mice, but I am definitely very curious about that. I mean, there have been analogous studies like to this that have been done, and so far, all the behavioral results have been inconclusive. But nonetheless, I am still very curious about it.
Well, I've done a little dive here as you're talking about some scary sci-fi scenarios, and show that in that initial paper, the statement is made that follow-up studies will clarify the relationship between altered cell cycle, increased brain size, and elucidate whether modifications in structural and behavioral traits exist. So I would guess that they don't act that different, or at least they don't act smart. But I would love to see those results. We got to get after the Silver Lab here and tell us, are they breeding our new overseers? For God's sakes, got to be some regulatory thing about that, right? Well, we're going to talk about some regulatory stuff in just a minute, which I'm excited about. And in the chat to the run up to this, the prep, I gotta say, like, not no BS here. This is a regulatory talk that you guys are going to be into because it's like a roadmap for how to bring your science into the clinic if that's what you're after, which many of us are. But before we get there, a quick message from Stem Cell Technologies. Are you busy in the lab? Aren't we all? Let Stem Cell Science News handle the literature search for you. With 20 newsletters covering immunology and cell biology research, they deliver the latest studies straight to your inbox. Subscribe by July 18th for a chance to win a custom lab coat personalized with your name. Sign up now at www.stemcellsciencenews.com/giveaway. All right, you guys, on today's episode, we have a special trio of scientists who are going to share the ISSCR's best practices for the development of pluripotent stem cell derived cellular therapies. This is an episode that we've had a lot of interest in over the months since it's been planned. And I would argue is the culmination of now decades of work, research, funding, conversations, hope, dreams. And now coming to a head and coming to fruition with the implementation, which has to be done very, very carefully. And that's what we're going to hear about today from Dr. Jacqueline Barry, who is Chief Clinical Officer at Cell and Gene Therapy Catapult. She's the co-chair of the steering committee and of the regulatory working group for ISSCR's best practices for the development of pluripotent stem cell-derived cellular therapies. Jacqueline Barry is a senior business leader with over 20 years of experience in biologics and advanced therapies. She's passionate about supporting the translation of advanced therapies and possesses a blend of expertise across the areas of regulation, quality, good manufacturing practice, science, strategy, commercial development, and management. So she's going to give us a lot of insight here today. We also have with us Dr. Kapil Bharti, who's a friend of the show. He's been on before to talk about his seminal work in the eye. He's a senior investigator at the National Eye Institute with the NIH, also co-chair of this steering committee and of the starting materials working group for the ISSCR's best practices for the development of pluripotent stem cell derived cellular therapies task force. We need a acronym or initialism for that, guys, but you're going to have to be patient for me while I say it a couple more times during the intro here. After this, we'll just call it the task force. How about that? Dr. Bharti's team performs translational research on degenerative eye diseases using iPSC technology. We've talked a lot about that in the show, both when he was a guest, also in some of the Roundup where we've covered his work. In the lab, they use this technology to develop in vitro disease models to study patient-specific disease processes and to set up high-throughput drug screens and develop cell-based therapies for retinal degenerative diseases. And finally, we have Dr. Jack Mosher, who is the scientific director for the ISSCR and the lead staff member of this task force that we're going to talk about today. So to the three of you, thank you so much for taking the time to be on the show with us today and for talking about this important task force that, I mean, you guys have been devoting many, many hours to over the course of the last many months. Can't wait to hear some of the details. Thank you guys again for joining.
Thanks for having us.
Yep.
Thanks for having us. Yep.
Yeah, nice to be here.
Yeah. So yeah, thanks for being here. Like Dale had mentioned, this has been in the works for a little while back now. Back in actually 2021, the ISSCR launched this, ambitions standards initiative to do something that we're all hoping to do in the stem cell field, right? Which is to elevate the overall rigor, reproducibility of stem cell research. And this is not just a problem that's exclusive to stem cell biology. Of course, we talk about reproducibility across biomedical research, but especially important for stem cell research because so much of our work is translatable and it has an eye to the clinic. So can we start things off? Perhaps you could walk us through how the idea for this initiative came about and what unmet needs you initially aimed to address with it. Jack, perhaps you could start with us.
Thanks, Arun. As you mentioned, the Standards Initiative has been really several years in the making. I think probably it launched around 2001 as an idea to really address critical gaps in stem cell research and its application. And the nice thing about being an international society is that we hear feedback from all over the world about where the needs are in the field. And at the time, and even before, this was a big one. As you mentioned, we started the Standards Initiative really with the first document on the standards for the laboratory use of stem cells, both pluripotent and tissue stem cells. And actually, that work that was published in 2023 was started way before then, not with the ISSCR, but with groups like the International Stem Cell Initiative, the International Banking Initiative, and groups like that that recognized the need early on when ESCs started coming online, that there was a need for standardization and quality and rigor. And so we, in large part, took a lot of the work, the legwork that they had done, and brought it together with a group of international experts and published a document that really outlines 5 critical areas on the use of stem cells in the laboratory. That brings us to a couple of years ago when we launched this part of the initiative. So we internally call it phase 2, but it's the clinical side where we're developing the best practices. We just use that term, best practices document, for the development of pluripotent stem cell based therapies. And the goal of this document really is to develop a roadmap for academics, for people inside the industry, also helpful for regulators to facilitate the translation of these therapies towards clinic, with the hope of really increasing the knowledge that's growing throughout the field, elevating the rigor and reproducibility, and creating a set of international principles and practices that we've brought together leading experts from across the world to help us put together. And that initiative is chaired, obviously, by Kapil and Jacqueline.
All right. Well, I guess that brings us to Kapil. I mean, I mentioned in the jump there about your leadership in terms of the research and moving the work forward in a specific, field. I call it a field. But it was, I don't know about point of the spear. It's early stage, I would say, in terms of the diseases that are being addressed. And that's the major effort here is we're talking about the culmination of this translational effort. When I think about that first phase, the research in the lab, a lot of times, I guess researchers were generally in space about the ethical implications or about what the boundaries were. And that first standards document has been essential. And I would say a beacon. I think when people talk about like what the guidelines are and what the boundaries are, they talk about the ISSCR guidelines because there was a gap there, Jack, as you alluded to, that had to be filled. Here, we're talking about a different thing where, of course, the ethical application and a balanced application, the economy, I'm sure all those things are in mind. But really here, it's almost like a technical roadmap, I think, because as research scientists, and I'm speaking for myself here, but I think this generally applies, we don't really have an idea. We come up with the innovation, maybe we think to the level like the IP, and then it's like, okay, somebody run with it, right? So this is a real, like a material roadmap, I think, for a lot of scientists who haven't really thought about it before. So talk about that a little bit, and Jack, you too, but Kapil, let's start with you. About what the main goals of this new resource, who is it designed for, et cetera?
Dylan, thanks. First of all, thanks for having me on this podcast. I must say, a huge fan. I should tell you that actually folks in my lab are a huge fan of this podcast. They often use it as a source of information. So thanks for doing this, both of you, Arun and Dylan.
It's our pleasure. Thank you so much for sharing that. That warms my heart.
So to your point, the need for this document, I often tell my own story exactly the way you said, Dylan, is that when we got the grant to take iPS cells, iPS cell-derived RPE to patients for macular degeneration, and this was in 2014, so almost 11 years ago, I had no idea where to start from. And so put together an IND. What does that mean? What does this document look like? So I go to the FDA website, download a guidance document for, at that time, for cell therapies, which was not for pluripotent stem cell therapies, but in general for cell therapies, cell and gene therapies at that time. And this thick book, and I started reading. After the 5th page, I want to pull my hair out because I don't understand a thing, because the language is so technical. I'm like, what am I going to do? We have the strict milestones that we have delivered every year. How are we going to deliver those milestones? So I think that's the audience that I feel really passionate about, that this document is going to address. All the newbies, all the researchers who have new innovations happening in the labs. They have made new cell types, new tissue types. They want to cure a disease, different diseases. You know, there are thousands of diseases we could address, and they don't know where to start from. So they are for me one really audience that would benefit from this document. The other, as Jack pointed out in his comments, is that people from biotech companies who are now taking lab technologies and taking them towards more commercial aspects of this, they will benefit from this. Even we hope that regulatory experts across different jurisdictions will be able to view this document and kind of compare notes, because as Jack mentioned, this document is jurisdictionally neutral. We have experts from all over the world who have brought knowledge, and Jack will tell you more about what their specific working group has done in the regulatory piece of it. So I think this document has huge value for multiple different people, and we hope it will be utilized even same like the previous document that ISSCR put out.
Jackie, why don't you elaborate on that as Kapil kind of offered? You have I would say, a more nuanced view. I wouldn't say on the ground, but maybe closer to that industry translation. And from your perspective, what are the gaps that are being filled here, and how is it going to facilitate the translation, kind of globally speaking?
I think you're right, Dalen. I think we do have a very translational focus at the Cell and Gene Therapy Catapult. And we work with hundreds of collaborators per year, and some of them very experienced and some of them less experienced. And I think it's you know, what Kapil said, and Jack, that people are knowledgeable, they're knowledgeable at the science, but actually when it comes to translating, what exactly do you need to translate these products? So that's why when we started speaking at ISSCR about producing this quite extensive document that covers everything from this, you know, picking your starting material all the way through to clinical trial, and trying to give people on the ground, pragmatic, realistic guidance, I just thought it could help so many companies. And we're talking academics, we're talking small to medium enterprises, but actually a lot of the large pharma are moving into this field, and a lot of it is new for large pharma as well. So we're agnostic about who uses it. We just wanted to make it as comprehensive as we possibly could.
And comprehensive it is. It's over 300 pages, right? It's very comprehensive. But that's great. That's great. We get all the details in there. But I guess following up on that, because it's so comprehensive, how do you recommend someone diving into it? It can be a little intimidating to approach at first. So Jack, could you talk a little bit about, for folks especially who are just starting in translational work or clinical development, what's the way to dive into this?
Well, that's why we especially chose a roadmap, an interactive roadmap, because you're absolutely right. A 300-page document, I'd fall asleep after 30 pages, I think. You know, so what this is supposed to do is allow people to have an idea, think, not sure how to do that, find the particular section that they need, and you're able to navigate down through the particular area of interest. So you might choose to look at regulatory requirements, and you might choose to look at what's required in the UK for the whole translational pathway. Or indeed, you might say, I'm looking to purchase some starter material, what's required in all of the jurisdictions globally. So you're able to cut and dice it whichever way you want to. And it allows people to, as their knowledge evolves, they're able to move further along the document or move further down into the document. So the interactive roadmap was something we were really keen to follow, and I think ISSCR have done a fantastic job of it, and it's working really well.
Yeah, if I could just provide a little bit of perspective to that, since the listeners are not able to see the document at this point unless they've attended one of the handful of presentations we've had. But when you open the document, so to speak, it's an interactive PDF. It's really a series of flowcharts at its highest level. So you can go to one of the 7 sections and see a process-based document with some decision boxes. And at which point, if you need more information on a particular decision in that particular section, you can click on it. It then takes you to additional information. So it's kind of acts as a funnel at a very high level. You see very superficial flow chart and process, but the more you need to know, the deeper down it takes you. And that's where we get the bulk of those 300 or so pages are additional information, links to regulatory documents, guidances, additional references, etc. It's quite extensive, but as Jackie said, we hope very useful and easy to use.
Mm. I'm so excited to have a look at this thing because, yeah, I mean, just browsing through the process there, I think is going to be really informative on just how you got to think about your research in preparation or in hope of actually making a translational impact. I want to go back to that first point I made about this phase 1, phase 2, because for me, again, the phase 1 was this beacon, right? And the whole idea of being a global resource, right, and a beacon, shining bright enough to provide guidance across all these compartments in working groups. It was a little bit challenged, I think, there because there were different kind of, like, I call it, there were different laws, right? I mean, there were certainly different, like, ethical boundaries at the very least, or different comfort levels, let's say, in different nations with some of those things. So while it was, I think, very importantly, it provided some boundaries that I think were agreed upon. There was consensus. It wasn't up to the ISSCR to enforce anything, right? And there was no whatever. It wasn't a legal document. Here, I think kind of the same idea, but here I think it's easier maybe to set a standard for rigor that can be globally adopted, right? And of course, so you want to make it like jurisdictionally neutral, right? But you don't have to worry about the legal element here, where you have to respect the boundaries of different cultures, right? Here, you can have something that maybe would be globally a standard, right? So I'll ask, how do you establish that kind of neutrality while also maintaining a strong standard? Was that tough? Did you have different views among the many different contributors to the working group, or was there generally consensus amongst the scientists about what level of standards should be applied? I mean, I guess pretty crazy to ask if there was a general level of consensus amongst any group, much less scientists. But Kapil, why don't we start with you? What was the experience like trying to establish a global and jurisdictionally neutral standard? Like, it sounds like smart, but really tough.
Dylan, you know this well. If there's anything that scientists will not agree on, is a consensus, right? So because we all have strong viewpoints and we all have our own experiences to share, and that's essentially actually became the beauty of this document, because people had such broad expertise with them, and we worked on this over almost 2.5 years, mostly through Zoom and through Jack's help in managing everyone across, I don't know, 6 different time zones, even more. And then recently we got together in Edinburgh, where for 2.5 days we debated on every bit of it. And gosh, there were debates, I can tell you. But they were good debates, I have to say. I think the consensus was reached in almost in every case, through the term I think we have come to use is shared leadership. I think everyone showed a kind of leadership and came together and say, yes, we understand that the definition of starting material may be different in the US versus Europe versus Asia, or how a bank is qualified to be GMP bank is different in different jurisdiction. But let's put an asterisk there and let's define that in as Jack pointed out, in the breakout of the flow chart. When you click on those little tiny dots with the numbers, it shows you more details in that document. So that's how we essentially actually were able to make it even a stronger document, where we had much more comprehensive information using everyone's experience from different areas and different backgrounds. So I think that having more debate on it, in fact, was more work to put it together, but it essentially made it a stronger document.
And can I add to that? I think I actually thought the consensus, because this was a large group, I think Jack was over 60 of us, it's a large group, very disparate group, but actually the group worked incredibly well together. And I think ISSCR have to be complimented for this. And Kapil, you and I have talked about this. it's actually brought together not only a document which we think will be really useful, but a group of people who can help each other in the the development and share information going forward, which will help develop the field still further. So I think it's not just the document, which I think is fantastic. It's the shared knowledge, the shared leadership, as you say, Kapil, which will go on beyond the document also.
Yeah, the amount of generosity, I think, in terms of information that the experts shared to create this, and the way they interacted was amazing. And they came away with this, document of principles and practices. But I do want to be clear of what it's not, and it's not standards. And we went back and forth with how to describe it and title it for a long time. And in a regulatory space, standards has a very specific meaning. And although there were many debates about, well, should we make recommendations or standards in the document, ultimately that's not its goal. It's principles and practices to help anybody in any part of the world move these forward. As much as some committee members would like, or maybe all of us would like, to have standards to make things easier for everybody, because from a distance it seems like things would be, common sense to make a single user bank one IND, for example, is a topic we just were going through by email recently. But that may be a project for another time.
Yeah, that's an important clarification that these are not standards. It's a best practices document. And you use the word amazing, Jack, to describe the document. I'd like to use the word miraculous because, as we know, scientists love to disagree. We agree to disagree. So getting all these folks from diverse backgrounds together in the same room, the same space to really hash this out is perhaps nothing short of miraculous, but a testament to your leadership as well. So a little bit more about that global community that came together to put together this document. Kapil, perhaps you could talk a little bit about how you went about selecting the contributors and ensuring, appropriate representation across not just industry, but also academia, regulatory communities. This is, of course, a global field with global translational initiatives. So how do you ensure that everyone is represented here?
So I can give a little bit history of how we all came together to put together the entire working group. And Jack and Jackie, please feel free to add the details I'll miss out. So I think 2.5 years ago when we met in Boston, that was the time we conceived the document. And it was just a handful of us, Jack, myself, Jackie, and some of the other ISSCR leadership. And then from there, we set up to have a first meeting a few months later. Meanwhile, we had selected steering committee members, and there were 7 or 8 of us. We all met for a day and a half in close to Boston. And there we started to have a high level kind of a outline of the document, how it's going to look like, and started to put together who were going to be the working group co-chairs. So as Jack mentioned, there are 7 working groups. So we had 14 co-chairs. Some of the steering committee members selected to be the co-chair, like Jacqueline and myself are co-chairs on 2 of the working groups. And then the working group members, of course, were also ISSCR members, but also spread across different countries and different areas of expertise, from manufacturing to preclinical, clinical, and regulatory. And then and amongst all of them, we made recommendations for working group members for each of those working groups. And we tried to go as broad as possible internationally and all other aspects of experience and knowledge. And then Jack was a person who did all the hard work, reaching out to each one of them and inviting them. And I think, as Jack mentioned, everyone was so generous with their time, and pretty much everyone agreed that we reached out to Jack. I can't imagine, I remember anyone who said no to this. But any details I missed out? It was really complete input from every single person who was part of the committee to start with.
I suppose one thing that I would add, I think you've captured it really well, Kapil. We also really wanted to capture everyone along the development chain, didn't we? Early scientists, industry players who have taken things through into clinical trial, or even more experienced industry players, some regulatory people. So we thought not only did we have to have a global input, but there had to be a depth across all of the fields. So we tried to select people along the development pathway as well, didn't we?
Yeah, and it's also a nice mixture of academics and industry professionals, right? It's not a bunch of academics putting out stuff that sounds good and seems good. It was really a broad mix of experience that made the document come together and as useful as it will be.
Yes, I mean, look, miraculous though it may be, I will say that there is something to be said for, the fellowship and goodwill afforded by a common goal, right? And I think that's what this was really about. We're all looking in the same direction, and we don't want to blow it, right? We've learned lessons from rushed work in the past, so I think we're being really careful about it. But as we're describing it here, I recognize, and importantly, right, we can't have everybody writing in notes. But I think the ISSCR in the past has been very good about inviting diverse perspectives. And here, and we're talking about how industry and academics got together, but also ISSCR has been great about getting feedback from the general community and being really acutely aware, I think, of the implications and the impact of the science on, not just the community, but like the political sphere, and being aware that it can't just be scientific voices that are heard. And again, going back with the phase one, that was obvious, right? Because there were some major ethical implications that needed to be respected and many opinions invited, kind of like your general IRB committee, right? You need the lay person. But here you wonder, do we really need the lay perspective here to weigh in? But I think it's important also to have that perspective. So was there a part of this process where you invited more of the global lay community to not necessarily weigh in, but to get that perspective, a consideration of that perspective?
Yeah, so we have sent it out after we had an initial draft that we were all happy with. We sent it out for peer review, actually 2 rounds of peer review. We spent really the better part of a year soliciting interest from our community, announcing this document and asking if there was anyone that was interested in reviewing it. We'd be happy to have them do so. And so over those 2 cycles of peer review, we had nearly 80 people from across the globe, across different disciplines, within industry, outside of industry, and asked them to review it, and got a lot of feedback. The largest part of that was addressed, as Kapil mentioned, when we got together in Edinburgh and took the draft that we had sent out and now incorporated and went through all of that feedback we got from that first peer review process. And as we've gone through the other rounds, we've incorporated that as well. And we're also, the document now is also out to review by regulators, which will be an important bit of feedback for the document. So to the best of our ability, we've tried to include our community to review this, because just like phase one, this isn't something we can impose on people. Really, the adoption of it and its use has to be because the community views it as useful and has input. And those are kind of principles that as a society, we try and incorporate in all of our documents. This one is no exception.
Yeah, absolutely. I mean, critically important considerations there. I mean, let's reflect a little bit on, the process, because obviously it took multiple years for this to go from concept to completion. And what surprised you the most throughout the journey? Was it, on the scientific side of things, logistical side of things? What was the biggest surprise that came out of putting this together? Maybe Kapil, if you want to start.
Biggest surprise, the size. I wasn't I did not know it was going to become a 300-page plus document. I think first time when we came together and we were discussing, there was this idea of a roadmap, like a flow chart based roadmap. In my mind, as a scientist, I was thinking more simplistic, review like document. But then when we started talking, we realized, yes, it has to be roadmap, it has to be flow charts. And then in Boston, when we met, that's where the flow chart idea really gelled, and all the different working groups came together. But then really, from formation of working groups to teasing out every single detail to the last T that we have now, I did not expect that we'll get to that clarity and that granularity of everything. What do you think, Jacqueline? And regulatory bit is amazing the way you guys have put it together. If you look at the whole document, it's even daunting at first, and then you can remove one or the other jurisdiction, and then you can simplify it. Go ahead, Jacqueline. You may want to add more.
No, I agree. It's a big, hefty document, which we couldn't have done without Jack's amazing work with us all. What do I find most surprising? I think how well you are able to navigate through the document. It's really quite easy. And again, that's been Jack working really closely with the developers. So I think that's been a pleasant surprise, a real pleasant surprise. And again, I'm just going to talk about people freely giving their time. There's been no competition. There's been no trying to keep some information secret because it'll move things in that field along quicker than in others. It really has been a global effort to try to accelerate the trial of these products and ultimately drive down the cost, drive up the amount of clinical trials, get them into marketing quicker and get them into patients. So I think that's been that real drive with that vision has surprised me, I suppose, how closely we stuck to that vision throughout the whole 2 years we've been developing the guidelines.
Yeah, I mean, again, as you guys describe it, I can't believe I'm saying this, but I'm really excited to browse this regulatory document. But it's as I said, it's a culmination of all this effort of the community. So to see it actually taking shape, I find that I'm really curious of what that actually looks like, what the roadmap is. But let's talk about, impact, right? If you guys, like, realistically speaking, what do you think, or what do you hope or expect that this is gonna, how is this gonna move the needle? Is it a matter of pace? Is it a matter of, decreasing the regulatory threshold by all these groups that are going to have to sign off on trials? Jack, why don't you talk about if you had to speak about what your number one impact point is when you're selling this? What would you say that is? Short, long term, whatever you're thinking.
Well, yeah, happy to give kind of some high-level perspectives, but really, Kapil and Jacqueline being out there on the front lines can address, I think, the impact that they see coming from this. But, what we believe will happen once this gets out and adopted is it will drive down costs through efficiency, drive down time it takes through efficiency, potentially increasing the pipeline of products, and potentially accessibility as cost and time and expenses come down. Those are very idealistic goals, but I would look to Kapil and Jacqueline for how they see this impacting those kind of on the front lines developing the therapies.
I agree with everything you've said, Jack. I think what we aim from this is to stop people making mistakes and to remove that area of uncertainty, whether it be regulatory, whether it be non-clinical testing, whether it be how to do a clinical trial, because they're costly. People run out of grant money because they've gone down a particular avenue, or they can't persuade their board because they've already tried something and it didn't work. We're hoping that this will remove those areas of doubt. I think that's what we really hope. And the other thing, which only when Jack was talking, I thought was, I also, everyone wants regulatory harmonization or convergence, whatever you want to call it. It may well go towards that. If you have the regulators understanding what's required for these products globally, and there's a community working towards it, then there becomes almost a driving force for that harmonization. And if you can harmonize requirements globally, then of course your time to come to market comes down, your cost, all of those barriers fall away if you've got more convergence.
Kapil, how about you?
One thing I will add is that it will reduce anxiety. I think for researchers who do want to take products to patients, it will definitely reduce anxiety, will increase the enthusiasm, like you said, Ellen, that you will feel that this is a document that you want to access. I have my coffee mug here says, "Keep calm and let the project manager handle it." And this the gifted by my project manager when I was always so anxious. But what about this part of the IND? What about the preclinical study? What about the QC? How are we going to do that? And now all of that is in this document. You can just click on it and you can find it. In fact, when we did the webinar last month, my part of my team listened to the webinar, and my project manager came to me at the end. They said, "Actually, after this document gets published, you don't need me." I said, "No, don't think that this is going to replace you." But essentially, that's the point, to have a guidance for the community. And we hope really this will be used as a guidance document. Guidance as best practices, not standards. We need to clarify that.
Important clarification, and shout out to the project managers out there. So wrapping things up, I guess more on the logistical side of the future directions here. Jack, perhaps you could talk about what's next for the standards initiative itself. I mean, we talked about what the impact of this document will be, but what do you think is coming next for the standards initiative? You know, are there future phases, complementary resources, all already in development? Because of course, the science is going to continue to evolve. So what is next for the initiative?
Yeah, I like the use of the word evolve, Arun, because both of these documents will over time evolve in the sense that they're not static. So going back to phase one, the laboratory uh, laboratory standards for the use of stem cells, that one will probably in another year or so be revisited. And at the time, we'll look to see what needs to be updated. But as a shout out to STEMCELL Technologies, which I know has some support of this great podcast, we are currently working with them for some video learning modules that will the community will learn about here, I think in June around the annual meeting. And we're looking forward to those coming out in, say, fall of this year. So that is part of the adoption process of the standards, which is really important. We believe in kind of elevating the rigor and reproducibility of the field. So that's phase one. Phase two, what we're talking about now, there'll be a big awareness campaign immediately, right, to try and elevate awareness of this document and increase its use. And with that comes feedback. And we've already discussed as a group that, just verifying the links in this document, which there are hundreds, will probably be something we need to do semi-annually as guidances get changed and websites get changed to verify that they're still accurate. And then probably every couple of years go through this document and revise it as needed. Future things coming forward from the society, there are a couple other projects kind of in line with standards initiative, though not formally underneath the umbrella, but a new disease modeling and drug discovery initiative that will be announced soon. I think will be in line with a lot of this work that is advancing the field and helping scientists such as you, Arun and Daylon, move your science forward as rigorously and easily as possible.
Well, while we're plugging STEMCELL Technologies, I have to highlight their tagline, or one of them, which is scientists helping scientists. And I have to say that I think we're all aligned. And this document and all the work that's been done by the many task forces led by the ISSCR are well aligned with that mission. I'm very proud of that. Of course, all this is provisional, and that's what's so great about it. Just like our knowledge of the natural world, it's not static, it continues to evolve. And while these standards may be evolving, they provide, I think, a roadmap, which to me is kind of a way of preventing, as you described, all the mistakes. It's kind of like the journal of unpublished results that we all dream about that's never going to come around, right? Is, well, why make the same mistakes that our peers have made before us? And one great way to avoid that is to provide a roadmap that is, as I said, provisional, but provides a really nice and instructive pathway towards translation. And that's what we're all so excited about. You guys have been so incisive in sharing the details on a granular level, the impact and the context for this task force and its product. And we really appreciate that. And looking forward to seeing you all in person, hopefully in Hong Kong.
Thank you, Arun. Thank you, Dylan.
Yeah, thank you both. That was great fun. Thank you.
Yeah, a pleasure to be on. Thank you so much for letting us use this platform to get the message out there. It's great, and great to be able to bring this to your audience.
All right, you guys, that brings us to the end of this episode. Pretty exciting for a reg show, if I do say so myself. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and roundup papers. You can also reach out to us on X at StemCellPodcast, Blue Sky at StemCellPodcast.com, or via email at info@stemcellpodcast.com with feedback or to suggest guests. Until the next time, for Arun Sharma, I'm Daylon James, thanking you guys for listening. Tune in